The data set comes from SciLife Lab in Stockholm, and they are working in one of the projects on identifying novel therapeutic antibodies. And part of the work then related to creating a library of lead candidates against a specific target. And to produce this library, they wanted to establish an efficient protocol for transfection and find out the optimal settings for transient gene expression with the Effector Pro transfection reagent. And as I said, this was the experiment was run using Ambr15, and the two factors that they could change was the amount of DNA that was added and also the ratio between this Effector Pro transfection reagent and the DNA, the amount of DNA. And the Ambr15, they were using 12 bioreactors, and with only two factors, that means that you have a very good optimization design. I will now go through this data set very much in the same way as I just walked through the mission popcorn. So the first thing that you always have to define is the critical quality attributes, what you can measure, the responses. And the main objective here was to find conditions yielding the highest possible transfection efficiency of this cell type. And SciLife Lab then has this knowledge that this would also encode or correlate with a higher protein titer. So the responses that they measured were in percentage were transfected cells and viability. And for the transfected cells response, 80% was seen as a very good result. And here you see the specifications that the transfected cells and viability should both be high, ideally 80% and 100% above a certain corresponding minimum setting. The critical process parameters, or the factors using MODDE notation, were, as I said on a previous slide, the amount of DNA added and this ratio. And here you can see how the two factors were changed. DNA amount was changed from 0.4, 0.8, 1.2 in 3 steps, and the ratio 0.6, 0.8, and 1.6, not with equidistant distances between the different settings, but that's another story. Here we see the 12-run worksheet where the experimental name, now using the MODDE Q for Ambr, the original run order is then replaced by the name of each vessel in a specific culture station in a randomized fashion. So this is culture station 1 and experiment 5. There was an experimental hiccup for them, so experiment number 6 had to be excluded because of unforeseen experimental problems. So it means that when we evaluate data, we're down to 11 experiments. You will get quality performance indicators of the data and the model, a quartet of bars for transfected cells and a quartet of bar of viability, and what very reliable results were obtained, very good modeling statistics, which means we can trust whatever maps or graphs we are looking at. We can look at the coefficients, and they show that there are some nonlinear dependencies for transfected cells. So the factors combine nonlinearly to influence transfected cells, and the factors combine nonlinearly also to estimate viability. It's easier to interpret what we call response contour plots. So to the left, we have the plot for transfected cells, and to the right, we have the plot for viability. And we can see how these two responses are predicted to change when we change the ratio of Effector Pro to DNA and when we change the DNA amount itself. And here we now see the minimum levels emphasized. Remember that we specified earlier on that transfected cells should be above 60 and viability above 80. And it is these lines are then emphasized by a slightly thicker line thickness. So here we have the minimum setting for transfected cells, and here we have the minimum for viability. Highest viability is here in the left-hand part, and highest transfected cells is over here. So they are conflicting, they are not harmonizing. So it means we must find a compromise where there is a low risk of failing to comply with the specifications of being above 60 and above 80. That means we can look at a sweet spot plot So where is the best operating condition? Which combination of the two factors encodes the best chance of fulfilling the responses, transfected cells being over 60% and viability being over 80%. So we can make this sweet spot plot, which would have been state of the art 7-8 years ago, or we can use more contemporary tools looking at the risk of failure to comply with response specifications. And the arrow here shows a low risk region where there is a low risk of being below 60 and 80%. The main conclusion was that SciLife Lab was able to set up a robust protocol while minimizing both the plasmid DNA and transfection reagent, and it ultimately led that they could lower the experimental cost. So the DOE really helped them to understand limitations of the transfection system and how to push the system towards the lowest use of raw materials. And also, as was accounted and told us, and that is also indicated in our hands-on computer exercise, they found Ambr to be a very useful system to work with.